Hello, this is Mrs. Ness-Deden, and today we are going to talk about the process of DNA replication. You've already learned about the structure of DNA and how DNA is made of nucleotides that are attached to each other by covalent bonds along the sides and hydrogen bonds across the middle. And you learned about some of the scientists who helped discover that. The reason that structure is so important, or one of the reasons, is that it allows DNA to replicate, which means to make copies of itself. So one DNA strand can easily break apart and make two identical copies of itself. This is really important when cells are getting ready to divide. As living things grow, their bodies need more cells, so those cells need to divide, and every single new cell needs a copy of the genetic instruction. So the DNA has to be copied before the cell is able to divide into two. This is a really complicated process that involves lots and lots of enzymes to help. If you remember, enzymes are proteins that help carry out chemical reactions and processes like copying DNA. And in this picture, you see many enzymes that are assisting with this copying process, and their names are over on the right. And one thing I want you to notice, we're not going to learn about all of these in this class, but I want you to notice that enzymes frequently have a name that ends in ASE. That's just a really good thing to notice because it can help you remember kind of what those molecules are and what their job is. So anytime you see something with a name that ends in ASE, that's most likely an enzyme. The two that we're going to focus on in this class are the enzyme called helicase and the enzyme called DNA polymerase. Those are the main two that we're going to talk about as we talk about this process of DNA replication. So we start with one strand of DNA. The first step in this process of replicating is that the two sides of the double helix need to be separated. And this is one of the reasons that the structure of DNA is so important. The enzyme helicase is the one that does this step. Its name can kind of help you remember what it does. The ASE ending tells you that it's an enzyme. The helicase part at the beginning sounds like helix, right? So helicase breaks apart that double helix and separates the strands. And the reason it can do it is that in between those base pairs, remember the bonds that you have right down the middle of a DNA molecule through here and here and here, that's where those weak hydrogen bonds are located. And so it's pretty easy for a DNA molecule just to kind of unzip right down the middle. And so the enzyme helicase breaks those hydrogen bonds and separates those two strands. Then once you have those two strands separated, a new strand can be built. So in this image right here, this one is the original strand that you started with. And so my writing might be a little messy here, but this is the original strand. And the one on the top is actually the new one being built. So this only shows you half of the original double helix. This would be happening again somewhere up here. And this one is the new strand that's being built. And you can see that big kind of beige colored oval back there is an enzyme. This step is accomplished by an enzyme called DNA polymerase. Again, ACE means it's an enzyme. Polymer, the first part of that word, remember, a polymer is a molecule that's a whole bunch of monomers linked together. So it's building something, and in this case, it's building DNA. And it does this by following the base pairing rules. The cool thing about DNA is if you know one side of the strand, you can build the next. And so these new bases are being added following those base pairing rules, and then they form a hydrogen bond right across here. And so you can probably predict what would be the next new nucleotide that would be attached right here. What base would it have? Hopefully, since you saw that this one was a G, you could predict that the next one brought in would be a C. And it would form a hydrogen bond with that guanine, and it would continue that backbone along here. And then what would the next one be? You have an A here. What would you have on this side? DNA polymerase would bring in another nucleotide, this time that had a thymine attached to it. to build that next little step. And it would just continue building that other half, following those base pairing rules, to recreate the other half of that molecule. The really cool thing about this is that because of those base pairing rules, because the bases always match up the same, in the end, you end up with two exact copies of the original DNA strand. So this one on the left is the one that you started with. We split it apart. The enzyme helicase splits it apart. DNA polymerase brings in the new nucleotides based on those base pairing rules. And then at the end, you have two copies that are exactly the same. They all have AT, TA, CG, GC. They all have that same pattern of bases on both sides. So this is a really important process. As cells start getting ready to divide and multiply and make more cells, every single cell needs to have an exact copy of its DNA. I hope that process made sense. Go in and read your post on Google Classroom so that you make sure you have done everything you need to for today. And I hope you have a wonderful rest of your day.